Welcome everybody. Thanks for coming along today, whether you're online or in person, and thanks to FGT for hosting us here today. My name's Michelle Buntain. I work for Tasmanian Institute of Agriculture. I work in the horticulture section and mainly in communications. This project, it was conceived, I suppose, and got up and running based on experiences from overseas. So the good results they were getting in Canada and New Zealand. So why don't we do it in Australia? That was the question, I suppose, and that's why the project's up and running. It's an IPM-friendly method of controlling codling moth, and that was another impetus for getting the project running. The project officially started in 2019. Now, that might seem quite some time ago, but there was this nasty thing called COVID that intervened, and that slowed things down quite a bit, particularly because it had international collaborations, this project. Now, as part of the running of this, there will be questions and answers, but if you think of a question and it's burning, just put it into the chat function if you're online, or if you're here, just remember it. Okay, let's get going. Okay, there's a big project team associated with this project. I think there's 7 different organisations, so well done to Sally for keeping everybody on track. And thank you to all those project members for what they contributed. And to our grower partners, they're integral to the project. So R&R Smith, Duggins and Bowden Brothers Organics. And a funding partner, Port Innovation. Today we have 2 presenters presenting exactly what's happened in the project. So we have Dr Guy Westmore from Department of Natural Resources and Environment Tasmania, and Dr Sally Bound from Tasmanian Institute of Agriculture. Now, our first presenter today is Guy Westmore. So, Guy, he's a senior entomologist with Biosecurity Tasmania and current chair of the Australian Fruit Fly Technical Advisory Committee. He's a national expert on the establishment and maintenance of fruit fly pest-free areas. Guy also works, his work involves invertebrate diagnostics, surveillance, risk analysis, and development of emergency response protocols for eradicating pest incursions. Today, Guy is going to tell us all about how, despite COVID, the team managed to secure import of live sterile codling moth into Australia, conduct quality tests, and release moths in Tasmanian orchards. So now I want to hand over to Guy. So Guy's going to share the screen.
Thanks, Michelle. Can you hear me okay?
You're loud and clear.
Great. I'm just share my screen. Okay, can everyone see my shared screen? Okay, so why investigate sterile insect technology? Codling moth is a key pest of apple in most growing regions. It has developed resistance to many pesticides in different locations. We do have mating disruption and biological controls, and these work to a certain degree, but chemical controls can adversely affect IPM systems. And there are also neglected orchards, plus roadside and backyard trees that provide reinfection points for immigrating moths. So codling moth sterile insect technique has been used in Canada since 1992 and in New Zealand since 2014. And codling moth numbers and pesticide use in the treated areas in both countries have been reduced by over 9%. For those new to this technology, when we talk about the success of these programs and when they started, it's often taken several years from the first application to reach the point of success that's claimed. It's not like an insecticide application that works immediately. Typically, sterile insect technique for area-wide management of an established pest will take a few years, potentially several years, to reach the maximum level of control that can be achieved with the technique. And it's not a silver bullet. Biological controls and occasional insecticide applications may still be required. SIT, and I just usually refer to sterile insect technique as SIT. I'll do that through the presentation. SIT has been used successfully around the world, going back to the 1950s, for both eradication of exotic pest incursions, including here in Australia with both Queensland and Mediterranean fruit fly on the mainland, as well as suppression or area-wide management of established pests. With codling moth, the sterile insect technique for codling moth in Canada actually started out as an eradication program, but was never successful and was probably never going to succeed. And so it morphed over the years into an area-wide management program to suppress the pest. So how does it work? Well, the SIT technique for codling moth involves flooding an orchard with large numbers of sterile moths. And the purpose of this is to reduce the chance of wild male and wild female moths mating. Wild moths mated with sterile moths produce infertile eggs, reducing the population and eventually infestation levels. It requires a very high ratio of sterile to wild moths, so occasionally an initial suppression of the wild population is sometimes required, and that will depend from site to site. Now, when they are available, releases of single-sex strains, that is, males only, greatly increase the efficiency and reduce the cost of SIT. But in the case of sterile codling moth, there is no single-sex strain, so we are releasing, releasing both sterile males and females. The sterile moths are mass reared at the Oxia facility in Osoyoos in British Columbia. And they are sterilised with gamma radiation with a radiation dose of 200 gray. Now, there is a trade-off between the radiation dose and the fitness, competitiveness, and longevity of the moths, with sterilised moths generally expected to have lower fitness and reduced longevity compared to wild moths, and the higher the dose, the more that is the case. Now, Oxia has experimented with this over many years, with most of the testing done between 150 and 250 gray. The radiation dose applied to the moths that we used is 200 gray, which completely sterilises the female moths and produces a high level of sterility in the male moths, so that very few viable offspring are produced from wild sterile matings. New Zealand, in their program, used moths irradiated at 150 gray, so a lower dose, which they determined to be the optimum sterilising dose for both fitness and sterility. But Canada has been using 200 gray. And as you can see from this diagram, these sterile moths can be distinguished from wild moths due to a permanent internal pink dye marker from the larval dye. I will show you a photo of that. It is. So while at first glance, the sterilised moths may appear similar to the wild moths, as soon as they are pressed, even just gently, they show the bright pink colouring, which is very distinctive. Now, after a week on a trap, the dye colour does fade a bit, quite a lot actually, but under a microscope, the sterile moths can still be distinguished from the wild moths. So there is enough residual dye there to separate them. This is just a couple of pictures from Osoyoos. On the right is the sterile facility, the radiation facility, and there are the orchards of the Okanagan region. Oxia produces about 220 million sterile codling moths annually, and they are released into the commercial orchards every week during the growing season, which for them is May through August. Mapping of the known and potential distribution of cotton moth was a small part of the project. The map on the left is a model that we produced of the suitability of cotton moth habitat based on Australian specimen records. And the map on the right is a published model using a global distribution record set. There are some differences, as you can see, but the hotspots in both models are generally in agreement. So we're looking at the central part of northern Tasmania, southeast Tasmania, southern Victoria, and in the area surrounding Adelaide. Now, the process to get the approvals for importing live cotton moth was a major part of the project. It consisted of two separate assessments by different areas within the one federal department. I won't go into all of the parts and the departments, because since then it's split again into different departments, and I struggle to keep up with them. And I think whereas those two approval processes might have been done within the one department, they would now be done by separate departments. And on the right there is the permit that was eventually secured. Now, Michelle mentioned that COVID caused a lot of disruption, and it really did delay the project, postponing for about 2 years. But in one way, this was a blessing in disguise, because it took 27 months from the time we submitted our applications to the time we received approval from DAF. And I think really that the reason for that is this was the first ever approval for import of live animals for direct release into the environment without first going into quarantine. So it was a groundbreaking process. It really has set a precedent, and it received a lot of scrutiny before we were finally successful. So obviously we have imported all manner of live animals into Australia before, but they have always gone into quarantine, whereas this was to import the moths and release them directly. So another major part of the project was investigating and developing the entry pathways for sterile cotton moth into Australia. And this slide shows the complexity of the supply chain to import the moths, which consisted of 3 flights plus 2 legs of road transport. You can see on the left, top left, that little red marker there, that is a Suez. So it was a Suez to either Kelowna or Penticton, then across to Vancouver, from Vancouver to Sydney, Sydney Hobart, and down to the Huon Valley in southern Tasmania. Now, there were 2 potentially viable pathways per week, but due to the cost of releasing on Saturdays, realistically, we only had 1 feasible pathway available, which was a flight from Canada with Qantas on Mondays. And that gave us a minimum 60-hour transport chain with releases taking place on Thursdays. The other complication, which was very unexpected, was that during the peak ski season in Canada from 1st of December to 31st of March, some of the domestic Canadian airlines and airports stopped taking live animals as freight so that ski gear could be given priority. It's obviously taken very seriously in Canada. So this resulted in changed schedules in terms of both airlines and domestic airports in Canada at various times. So this is how the moths were packed up for transport. Each cup contained, they're little sort of paper cups similar to those old style ice cream cups. Each cup contained approximately 3,000 moths, and we imported about 90,000 moths each week. Now, the cups were insulated inside the boxes, and the boxes contained ice bricks to keep the moths cool and dormant. And the bricks themselves were separated from the cups to prevent chilling injuries. So the moths essentially were dormant. They didn't move once they were packaged up and cooled. So they needed to be kept in a cold supply chain for that full 60 hours to reduce damage. The next few slides look at the testing we did to determine whether the moths were in good shape to be released and do their job. And we did this by looking at 3 separate consignments through the season. We looked at male to female ratio, weight, longevity, mating ability, and flight ability. The first step was to separate the moths into males and females. To do that, we used the external genitalia of the moths located at the tip of the abdomen, which we could see under a microscope. And for each consignment, we separated about 600 moths for the quality tests. So these pictures here show some of the experimental setup. The pages where the moths were placed for the tests. Some of the tests were straightforward. The mating ability test was a bit more involved, where for each female moth, we had to dissect out the internal genitalia and determine whether the male moths had deposited spermatophores in the bursa copulatrix, which is the part of the female genitalia used to store the male sperm. Now, in an ideal world, we would have tested the mating ability between the sterile males and wild local females, but we don't have access to a population in culture to do that. So we tested the wild males with the wild females for the same example. Here are some of the results from those quality tests. The average weight of the females came in at 30.8 milligrams, which was just above our expected range of 25 to 30 milligrams, so that's quite good. The average weight of the males was 17.7 milligrams, which was just below our expected range of 18 to 20 milligrams, so not too bad, almost there. The sex ratio, male to female, was pretty much as expected. Mating success was quite good, noting that these tests were under control conditions where males were only given access to females for a short period of time. And longevity was also more or less as expected, about 2 weeks, with female mortality slower to kick in than for males. And not surprisingly, the moths of both sexes survived better when given access to water. So half of our treatments had access to water and half access to water. This was a secondary trial we did to determine what the effect was of any delays in the transport chain. This was done after we discovered that delays were a very real and problematic occurrence that we had no control over. I'll talk a little bit more about that later and the effects, but what this particular trial shows is that we could get away with a 24-hour delay with no significant difference to the moths that were released on time, but anything more than that and mortality rapidly increased. The moths were released at 3 trial sites paired with controls. The sites were approximately 4.5 to 7 hectares in size. This is very different to the 200 plus hectares that New Zealand used as a pilot trial. But again, that was really to determine the actual effectiveness of the sterile releases, whereas we are at a much earlier stage in this process, I guess you could say. We're trying to determine the feasibility and economics of importing and releasing moths. The release sites were chosen to get as much isolation as we could from the expert apple blocks, with buffers of non-host vegetation where possible and minimal pesticides. The moths were released most weeks from late October to end of February. They were meant to be released every week, and I'll get into that a bit later, but most weeks they were released on a weekly basis. And the release rate was approximately 6,000 moths per hectare, which is obviously works out at a tad under 3,000 male moths. Now, two of the sites worked very well because they were paired with controls on the same orchard. So they had very similar levels of pest pressure and the same management. The third trial site in retrospect was not good because it was paired with a site on another orchard with a different pest pressure, different management, but we didn't have a lot of choice in that, so we got that. Release methods weren't part of the project. And so because the sites were very small, it was simplest and cheapest to release the moths by hand each week. So that's me on the right, and on the left is Ramandeep, who was one of our casuals who also did one of the releases. Now, these photos weren't part of the project. They are from a separate visit several years ago to New Zealand that Ian and I were involved with. And this shows the two main methods that have been used in New Zealand orchards to release sterile moths. On the left is a hexacopter drone, and on the right is a fixed wing drone. And in Canada, they are also using ground releases via ATV. Every week, the traps were checked to count the numbers of wild and sterile moths. So Penny on the left and Ian there in the middle. And that is an example, that sticky trap on the far right. And I can't remember off the top of my head what red and blue corresponded to, but I'm assuming red was the sterile moths and blue was the wild moths. In February each season, there were also field assessments of damage at the release at the control sites. Okay, so even though we weren't really testing the effectiveness of itself of SIT, just because that requires a much larger area and it requires really a multi-year project, we did still record the data and have a look at it. Long-term codling moth trapping data from across Tasmania typically shows two large wide peaks of activity in mid-November to early December, and again in mid-December to early January. Although it is variable from year to year, of course. And these correspond to the emergence of early and later cohorts from the overwintering run. And this certainly shows up in the wild captures at both the release and control sites, as you can see from these charts here. The results are mixed. Again, with the very small release sites open to immigration from surrounding blocks and only 2 seasons, we weren't expecting to see any success at this point in time, but I think it is still very interesting. But look, there are 2 key points here, I think, in terms of the feasibility of this as a long-term program. One is the low trap numbers of sterile codling moth in spring, which is a concern, but this is something that has also been seen very consistently in Canada as well, and the program still appears to be successful. The other bigger problem is that in both years, we were not able to release at a key period of moth activity in late December and early January. Because there was only one, we only had access essentially to one flight out of Canada on the Monday, each Monday. Any Canadian public holiday on the Monday resulting in a cancelled shipment. And in the 2 years where we conducted the releases, in both of those years, we had the Christmas and the New Year public holidays either falling on a Monday or the public holiday in lieu for that public holiday. So that resulted in a 3-week gap between the sterile moth releases at a very critical time during the season. So this is the first paired site. I hope the charts are fairly self-explanatory, but more in fact to talk about when we get to Q&A later. You can see, I hope you can all see my cursor here. You can see this peak here of the wild moths, November going to December, and again, secondly, December coming to January. And this area here, where we've got these 2 red arrows for this 3-week period of no releases, that is where we would have hoped to see a much higher peak of sterile moths in the orchard. Nevertheless, fruit damage at the release sites was much lower than sites. The second site shows a fairly similar pattern to the first, although without the large peak of wild moths at the site in the second season. And again, in that second season, we had essentially three releases. Sorry, three periods where we didn't release. Again, probably not significant, but the release sites have lower damage. Now, this third pairing, as mentioned previously, was very unfortunate. We had very different pest pressure management on both sites. The release site, well, the control site was at Codling Moth under very good control. There were very few wild moths detected at any time during the season, whereas the release site had really very high levels of wild moths. And as I said before, sterile insect technology really requires a low moth population to begin with to be successful in a low insect population. And that really sometimes requires a knockdown insecticide application at the beginning of the season if there's a high background pest pressure. We were limited by options within the growing area that we'd chosen. We wanted it all to be in the Huon Valley to minimise the travel time and the cost to the project. But I think if we had our time over, we would have searched further afield for a pairing within the same orchard, or potentially even just stuck with the two sites and released more moths at those sites. Okay, this is a summary that's been put together by Michelle, but I just wanted to just go over a few of my own conclusions before handing over to Sally. My interpretation of these findings is that in an ideal world, if we could guarantee security of supply through the season, with no cancellations and no delays, then it would be technically feasible to import sterile moths from Canada for release in Tasmanian orchards, not taking into account the economics, just in terms of the technical side of things. Sally is going to turn on that in a moment. The moths are still viable and they appear fit enough to be released after 60 hours of transport, but there isn't a lot of margin for that. The low trap numbers in spring are a concern, but again, this is something that's been consistently seen overseas, where I think they tend to manage it with a single insecticide application at the start of the season. And the program as a whole still appears to be successful. However, so that's really in an ideal world. The problem that we experienced was significant disruption to the supply chain each season, and it looks to me like this is a permanent feature, making it too unreliable to rely on the importation of moths from Canada. So I think if industry does choose to go down this path, then I think the only choice would be to produce the moths here in Australia, or potentially even import moths from New Zealand if a facility ended up being built there. But I will hand over now to Sally, who is going to talk more about the economics of the sterile codling. Thanks.
Thanks very much, Guy. And Guy's involvement with this project hasn't stopped, because I think on Wednesday he gets on a plane and visits Canada and visits the production site and looks at how they're actually releasing them in the field in Canada. So thank you very much, Guy, for all your involvement.
But Michelle, well, can I just jump in and say that trip, people might just be wondering what's happening at the end of the project. I should just mention that that trip was actually booked for March 2020, and it was literally a week or two away when the borders were, a week or two before that when the borders were closed and the trip was cancelled. And this is actually the first time we've been able to reorganise it. So
we didn't want to leave Guy stranded in Canada, so we didn't send him. So we keep him here in Tas, so it was very good he didn't get on a plane. Otherwise we may not have seen him for 12 months. Anyway, we're going to hand over to Sally now. And I'm going to introduce Sally. Dr. Sally Bowd. Many of you know her very well, but she's a Senior Research Fellow with the Tasmanian Institute of Agriculture. She's recognised within national and international research communities as a leading researcher in pome fruit crop load management. Her research focuses on crop manipulation, fruit quality in apples, pears, and more recently on sustainable orchard floor management. Sally is the project lead, so she's had to contend with all these issues of COVID and moth shipments not coming in when they should, and just the timing things. I think project management was really important for this project, and well done, Sally. So she kept it on track. Today, Sally is going to cover off on the all-important economic aspects of sterile codling moth program for apples and pears. Thank you, Sally.
Thanks, Michelle. I would just like to point out that even though I was project leader, I did have a wonderful team that I couldn't have done this project without. Okay, so as Michelle said, I'm going to talk about the economic aspects of this project. Guys covered all the technical parts of it. So towards the end of the project, we commissioned Big Finger to undertake an economic analysis for us on the viability of sterile codling moth in Australia. So this first screen basically just shows the costs of actually importing. So we have, get this mouse working. Hang on, I've got it here. So as you can see, the sterile codling moths are less than a third of the total cost of importing. So if we can get the cost of the moths themselves down, then we'll find that this is viable, but I'll cover that shortly. So freight actually tripled in price from the beginning of the project when we did our initial project costings to when we were actually starting to import our moths. So that really blew our budget out. And then, of course, we've got our quarantine and border force fees and our import charges as well that all add on. This is a comparison put together by Nick, and it is using data from Canada from a paper by Gill back in 2014. So it shows a comparison between the different strategies. So we've got our pesticide control, mating disruption, and sterile insect technique. And what Gill reported was the degree of damage with each of these methods of codling moth control. So as you can see, we had reasonably high rate of damage for the pesticide control, much lower for mating disruption, and even lower for our sterile insect technique. and that represents quite a considerable fruit value loss. So we're going from over $2,000 per 50 tonnes down to 307 for our mating disruption and right down to $30 for our sterile insect technique. So you can see there's a big difference there. So if we do a comparison relative to the pesticide-based control, we find that mating disruption comes out well above, but our sterile insect technique comes up better than that as well. So it is certainly a practice worth doing if we can get our costs sorted out. Nick also undertook a range of scenarios looking at pesticide application, mating disruption, and sit at a range of different costs. So if we can sit down to $750 per hectare for our releases, then we're really laughing. It's certainly feasible. Even at $1,000 per hectare, and at $1,500, it's just slightly under or almost breaking even with mating disruption. So our net present value, based on 8% after 20 years, based on these costs, overall, you wouldn't be planting an orchard these days, but we can still use this to compare the value of the different control methods. So as you can see if I can use the mouse here. So our cost for pesticide application, or with pesticide application, is nearly 500,000, and that does reduce with mating disruption, and significant reduction there with our sit at the lowest costing, and then at our highest costing, 1,500 per hectare, somewhat similar to our mating disruption. So if we compare it relative to pesticide only, we're getting a much more positive value with our $750 per hectare for the sit. And again, that again shows that relative to pesticide, it's very similar to mating disruption. And if we compare it relative to mating disruption, you can see that we're coming out on top at the two lower rates of sit costings. Okay, so let's move on to the benefits of sit. So there are additional benefits apart from direct dollars and our direct reductions in fruit damage and associated increases in gross return. As Guy mentioned, we get reduction in chemical usage, which is very environmentally beneficial. And British Columbia have reduced their pesticide use by 96%. Since they first introduced it. It didn't happen straight away. It has taken several years to get to that stage, but it is a significant reduction in pesticide use. SIT is an environmentally safe technique. It provides for residue-free pest management, and it avoids the problem of pest resistance being developed. It is a species-specific technique, so there's no effect on non-target organisms. So there's no damage done to our beneficials, so we can keep building up our populations of beneficials. It's readily integrated with other techniques such as mating disruption and granulosis virus. It has area-wide management potential, and there are other benefits for other crops. Other crops that are hosts are quince, walnut, stone fruit, and then there's all the residential properties with their backyard apple trees that currently provide a real infestation point for orchards if they're close by. So the conclusions that we've come to are that SIT is a viable tool for codling moth control, and it can be integrated into an IPM program. As Guy said, it is technically feasible to import sterile moths from Canada, but it is logistically and economically challenging. And that's due to the lack of consistency of supply. There were some periods where we could not get moths into the country, and they often coincided with our second peak here in Tasmania. Long transit times, which significantly reduced the viability of the moths. and the high freight, quarantine and border force costs, which really puts it out of reach. And economics are also driven by existing levels of damage. If you've got high levels of moths in the orchard, then you need to reduce that before you can introduce SIT. So if the SIT moths are available at an affordable price, it is feasible to release our sterilised moths in small areas. In other words, in individual orchards. And what our pilot 2 years has demonstrated that. So we can reduce both pest pressure and damage. And there are some opportunities for cost efficiencies if we move to area-wide management, but that's more suited to really large growing areas, and there'd be very small regions of Tasmania where you could do area-wide, but it is still feasible on an individual orchard basis. The other thing that needs to be sorted is the technologies for releasing the moths. In Canada, they're currently doing it by vehicle. They have specialised equipment set up on ATVs and they just drive through the orchards. New Zealand have been experimenting with aerial techniques. They've used fixed wing plane, but now they're looking at moving to drones. But that's probably only feasible for area-wide management with quite large areas. The other thing I probably should mention about area-wide management is that to be successful, you need considerable consultation and collaboration between growers, local councils, and even the wider public for it to be effective. And that is something that will take probably many years to get together. So where to from here? Well, this project is now finished, but there is scope for further work to be done under another potential project if anybody's interested in continuing this work or if industry is interested in following up. So what we need to do is examine the feasibility of production of the sterile moths here in Australia. There's talk of multi-species facilities, which might help with cost reductions. We could potentially partner with New Zealand to produce moths. So either we produce here and offset some of our costs by sending moths over to New Zealand or vice versa. And we also need to ensure that we've got the capability for mass rearing of the moths. The economics and logistics of the potential release methods. There are differences between ground and aerial. Ground is potentially cheaper, but the larger the area, the more expensive that becomes in labour. So in that case, you might want to revert to aerial. There is better moth recapture with aerial distribution, or at least distribution high up in the trees rather than down on the ground. But that's another thing that should be looked at. And the other thing is, how are we going to fund a SIT facility for codling moth? That'd probably be a real challenge. But in the interim, if growers aren't using some of the more complementary IPM management techniques such as matting disruption, granulosis virus, and potentially mastrus releases in the future, then there's certainly scope to be moving towards that until we can potentially get SIT up and running here in Australia. My hope is that eventually we can do that. So I'd just like to reiterate my thanks to the growers. Without their input and their willingness to allow us full access to their orchards, we would never have achieved what we've achieved in this project, and to Hort Innovation for funding the project. So I'll pass back to Michelle and we'll move on to questions.
You want to keep the floor there, Sally? And I can pose questions from here if everybody can hear me. We'll start with...
Can you be closer to the mic to the online people?
Okay, come and join Sally. We'll start with questions from online people, and because you've got beers coming, you can wait. Okay, first question relates to the cost of setting up from Michael Crocera in Victoria. What is the cost of a setup of a SIT facility in Australia? Any ideas? Anybody?
If Peter Crisp was online, he could potentially answer this.
That's what's over there. No, it's with Michelle.
Oh,
Okay, Peter Chris, let me see. I'm not sure, we might let Peter put that in the chat if he's online.
yes. okay.
Otherwise, it might be a question we get back to the group with.
Yes, okay, sounds
Okay.
good.
Peter, if you're out there. Okay, this might be a question for Guy. Guy, are you online there?
Yes.
This is from Martin Van Helden. How did the QC results after transport to Australia compare to Canadian QC results with no transport?
Yeah, good question, Martin. The idea of the trip to the facility in 2020 was to look at the, I guess, the QC tests they were doing and try and sort of replicate that here. We didn't have that opportunity, unfortunately. And so we don't know whether our tests are, I suppose, comparable to the Canadian QC results. But I'm going to be, that's very much I suppose, the main purpose of the trip. And I'll be finding that out next week. And that'll go into the final report as well. So there's still a bit of information to come.
He posted on APAL's magazine to Australian Fruit Grower. We had an article coming out there and we might address some of these questions in that. A second question around transport. Do you think it could be improved, for example, by transporting in hypoxia, increased CO2?
Yeah, so I mean, I was a bit general with the ice bricks, so dry ice, so it does provide that high CO2 level in the packaging.
Hope that answers your question, Martin. If not, just put something else in the chat. Okay, one last question from our online peoples. Producing sterile, this is again from Martin. Okay, producing sterile moths in Australia would require building sterilisation equipment, staff, and a minimal market for sterile moths. What is the economic picture there?
I think, Michelle, that's very much an open question in terms of, it's obviously not something within this project that we could work out how much it would cost to set up a facility on the mainland for that. I suspect it's something that would have to be done in conjunction with New Zealand, as in the facility would have to be in one of the countries, and the cost and benefits spread across the two. I have heard people sort of suggest that the facility could be set up close to the Steritech facility in Melbourne, such that we would be able to produce the moths, but then the sterilisation component of the facility is essentially a commercial provider where we mass rear the moths and then almost send them next door, so to speak, to get sterilised before transport. So it might not necessarily be a full. We might necessarily have to set up the irradiation facility. It's more about mass rear.
And this question is from Duncan Park. Have your market access team negotiated SIP as part of a systems approach into markets, for example, Japan? I don't think we have a market.
No.
I can say no. It's
just on that, if we
not market access, but I don't think that's.
just, I guess one of the questions with that though is there any of the benefits, Sally, from that point of view? So when you want to go back into the cost benefit a little bit, but there was no looking at the benefits that might come from if we could reduce codling moth trapping or monitoring. None of that was built into a cost benefit. No, it wasn't. There's still lots that you could do, but the project had a finite budget. Yeah, sure. Yep.
This is from Michael Crucera. Are sterile codling moth releases viable in large areas with higher populations than Southern Victoria and Terrace? Ellie, do you want to address that one?
So
Okay, sorry, Guy.
So codling moth releases, I just can't see anyone in the room, so it's hard to know whether I should speak. But codling moth releases certainly are viable in large areas, and in fact, the larger the area you're talking about, I guess the more financially viable it becomes. But in terms of higher populations, no, sterile insect technique doesn't work when you have a high background pest pressure, and you really need to knock down the pest pressure first before you begin a sterile program. And once you've done that, you can use the sterile insect technique to maintain a low pest pressure, but you can't, well, it's very difficult to use just sterile insect technique to go from high pest pressure down to low. You kind of need to knock it down first and then maintain it at a low level with sterile insect technique.
Some of these questions may have come in after you explained that during your talk, so, but that's okay. Now we're on to Kevin Dodds, who, do you think we're likely to be using SIT and Mastris in combination in the future? This would change the economic analysis. What are your thoughts on the economic viability of a combined program?
Well, not being an economist, I find that sort of question very difficult to answer, but we could certainly put it to Nick Finger, who did our analysis.
And we might also put it to the team working on Mastris.
Yes.
Yep.
Yes.
Okay, good question. Last question from online is from, I can't read that actually. Anyway, my understanding is that in Stanthorpe, for example, growers have switched back from mating disruption to pesticides, albeit much softer ones, and they're using pre-mating disruption. Would that risk also be a consideration for SIT?
As in the risk of softer pesticides.
Yeah. Is that a problem for your sterile moths, having soft pesticides being used?
Well, if they're detrimental to normal codling moths, wild codling moths, then yes.
Yep. I hope that answers the question. If not,
I can add something there, Michelle.
excellent.
So one of the significant pests would be fruit fly. And Tasmania has the benefit of not having fruit fly, and the same with South Australia. South Australia does have fruit fly outbreaks at the moment, but they are attempting to eradicate those. So I think sterile insect technique would still work very well with in areas without fruit fly. Once you get into an area with fruit fly, if you are, I guess it depends on the location and the species, but if you are dealing with fruit fly as a secondary pest, then it probably makes sterile insect technique for codling moth a lot more difficult, because you are going to be applying some type of treatment. But again, it depends on the type of treatment, whether you're using baiting or male annihilation versus cover sprays. So I guess it really is going to vary, but certainly much harder, I would say, if you are growing in the presence of fruit fly compared to a fruit fly pest-free area like us.
That's great. Thanks, Guy. And now over to questions from the floor. We've got a number of people here in person, so anybody?
One for Guy. You mentioned that having more males SRT release made the treatment more efficient. Did having that sort of 50 to 60% female release inhibit this trial, in your opinion, or is that standard in Canada as well?
Oh yeah, there's no genetic sex and strain for codling moths. So wherever it's released, it's always males and females together. I was just drawing the distinction more to some of the other insect species like Mediterranean fruit fly, where they've developed a way to separate the sexes and release only males. So I wouldn't say inhibiting, but it's more that you are, I don't know if you'd be halving the cost, but you're certainly, for the same production of moths, you essentially can double your ratio, or you have the same ratio at half the cost. So yeah, it's not a relevant factor for codling moth at the moment.
Michelle, correct me if I'm wrong, but on the figures that are on the board, I think year one was cost per hectare was at about $6,000. Year two, about the same or
A
nine?
little bit higher.
A little bit higher.
Yes.
So for 100 hectare, if we've done 100 hectares, in year one, it had been a $600,000
Yes.
exercise. How do we get it from $6,000 a hectare to $750 a hectare
Well,
and
remember,
still make our SITS facility viable?
yeah, well, remember that a lot of the, 2/3 of that cost was actually for importing, the
Yeah.
freight and import fees and stuff. And I know there's a cost for SIT, which is why perhaps if we could go multi-species or something like that. But we have to find other ways of funding it.
So the site in Port Augusta, which is doing the blowflies and
Well, I think.
so that's wouldn't have the potential to be able to do those as well. I really like the idea of having them close by the radiation chambers. That to me that's very smart. But the facility in Port Augusta to me seems to be the only potential in the future without having a standalone facility.
Yeah, well, that's something that needs to be investigated further. It's not within the scope of this project, unfortunately.
And do we know if when we're doing the trials, if any orchards used pyrethrum at any stage? And would that also knock out your sterile moth population as well?
Yes, I'm sure it would knock them out. I haven't followed up about use of pesticides that are soft or whatever, so.
I mean, I'm saying that from an organic, where you can use pyrethrum, but obviously it's fairly lethal brood as well. I was just interested to know on the gauges that you had, was any of the sites having pyrethrum slotted in anywhere, which would have really knocked numbers around at a certain period of time, especially if you just done a release.
Yes, it's probably something that's worth following up if we get an opportunity. One thing that we haven't mentioned is that the releases were affected by weather. Wet weather certainly reduced the numbers caught in traps. And so the drier the warmer the weather is, the better it is for the moths.
And I know this question is probably out of your range, but when they're doing Biosecurity is doing the moth trap counts, how do they distinguish between the sterile moths that are caught in that trap and the wild moths that are caught in the trap? Do they have the technology to know if you squish them, they're big?
Yes, go ahead.
Guy?
Oh, thanks. Yeah, I was going to respond to something earlier, but just on that, look, if it came to an issue where we were releasing moths on, say, export orchard, yes, certainly the biosecurity inspectors would be trained up to distinguish wild moths from sterile moths. And certainly as part of the project, we consulted with and talked to the biosecurity operations team to make sure that they were aware of where we were releasing moths to make sure that there were no sterile moths being recorded as wild moths in export orchard blocks. What I was going to say earlier was, I think there was a question directed to Sally about the cost of the facility and how to get the costs down. One unusual aspect of this program is that there is, I don't know the extent of it, but there is a fair degree of external subsidy for the program in Canada. So there is some type of, again, I don't know the details, some type of poll tax or land tax or some type of rate, council rate potentially, that is levied on all residents in the area because it's seen as something that provides a benefit to the whole region. And so that essentially subsidises the cost. So I'm not saying that's something that needs to be done, but just be aware of that's one of the things that makes it very financially viable in Canada anyway.
Just another one for you, Guy. Just in the two sites where we had the control and treatment blocks, how far did you have an idea of, did you catch any treated moths in control areas and had an idea of how far moths can actually go?
No, not that I saw. No.
No, I'll answer that too. We didn't see any on the traps that we collected in county.
Yep. Thank you.
So just on the export one, so they were picked up Scotland. So if it did breach the threshold, they need to go back to entomology and then they could test whether or not it was a SID or not. And that was all approved back to DAF in Canberra and explained to the inspectors, the international inspectors as well about that, so that wouldn't compromise. We were a bit concerned about that. So what are your costs? And so the main thing was about damage. But so overall, firstly, given it seems that the mating disruption was about as beneficial as SID. Is that right?
Yes, at the highest level of SID costing it was, yes.
In terms of the comparison about, so the benefits in there, you talked about damage.
Mm-hmm.
So it was damage and the cost of it was just damage that was the benefit there, not the cost of not having to have as much insecticide. It was just damage.
No, it was just the damage. So the damage for the mating disruption was about 0.4 and for SID was 0.04.
Yeah, and which was significant. They were both in a similar region, it looked like, and very different to the insecticide option.
Yeah.
But there was a saving on both of those two because you didn't have as much.
You didn't have the pesticide application, yes.
But that wasn't taken into wasn't a net cost because the cost of SID was 6,000 hectare, but you saved on insecticide, is that right? Was that taken into account or not?
I'd have to go back to Nick on that and clarify that.
Yeah. I think it'd be really good to I don't understand the science bit, but maybe the numbers. But it'd be really good to be able to set that out to be very clear. Is it your plan to release a draft report and seek comment?
The report is actually overdue to our Hort Innovation.
Yes. So
there is option to send it out. I've had some feedback from the team members, but I wanted to get the report basically right before I released it to others. So potentially, yes, Peter, we could release it to limited people initially before we submit it through the Hort Innovation.
I guess the main reason being so is the next steps. So how far does this realistically take us, and what do we need to do next? And I guess that comes back in part as well, asking then. So how's it set us up to work out next, and how do we get the most use out of the work you've done? To put that line aside to say, okay, here's the next steps. Here's what we know. Here's what we don't. Here's what we could. Where's New Zealand at now in terms of what they're doing?
They have put it on hold since COVID. They had no shipments in during COVID, and at this stage, Guy might be able to expand on this, I'm not sure, but at this stage, they don't have any ongoing plans, but they are still interested in the technology.
Guy's shaking his head, saying no, he can't expand on that at the moment.
Yeah, the New Zealanders were actually subsidised by Canada as well with their imports.
Yeah.
So their costs were lower than our costs.
Yeah, and I guess look, the world's changed on freight again. Interesting to see how that really looks at. Are the New Zealanders then tracking how much their pest pressure is now?
I presume they are. I haven't spoken to anyone for a while from New Zealand.
They have the beauty too, Peter, of flooding a very high density orchard area completed. Yeah, which gave them, it would be like if every orchard in the Huon Valley participated. see a different result, I'm sure.
And they have the benefit as well of their export MRLs are quite strict as well, which also feeds into looking at technologies like this, make them a bit more viable.
I think Bob had a
Yeah, I
question.
have a question related to non-target insects, or non-target pest problems. Were there any benefits to in the management of them from this use of the sterile insect technique?
Because it's species big,
Yeah,
then it only controls the codling moth. It has no effect on other pests. So you would still need to control other pests, maybe by chemicals, maybe some other way. So that can again impact on the sterile codling moths.
Maybe a grower could comment on that.
Well, it's obvious that this technology is purely for codling moths. You still have your other issues, Bob, and conventional growers would handle those a lot differently to what, say, the majority of us here are organic growers. And we still have those specific pests and bugs are still an issue. But in saying that, nothing comes close to the issues that codling moth causes.
Question for Michael on the floor.
So mine's, I think, partly to the growers in the room as well. The economic analysis that was presented was based on GIB, which is Canadian data. And the level of damage of codling moth fruit in that seems quite low, even under a spray program. Obviously, if you weren't looking at 3.1% losses, but you're looking more at 5 or 6% losses, those savings from reductions would start to add up. Is 3 to 5% losses even representative of what the industry is facing, or
Like 5 to 10, Michael.
so that would kind of suggest that the economic savings could be even, well, probably understated by that economic analysis. If you can bring it down, instead of 3%, you can bring it down 7 or 8.
Is that your 5 to 10 in your situation, Scott? When
Yeah, I'm not speaking commercial.
No.
That's
Okay, take the side.
that's how that is.
Because insects are very effective.
Before they can at the right time, when insecticides are very big, no question about it. Just purely from an organic perspective, and every organic farm's different. Some may use a bit of pyrethrum, some purely relies on the grandex or the viruses. Some have mating disruptions, some have their own ties. But in our situation, we look at 5. We were prepared for 5 to 10%. And the reality is the virus, when you read the grandex label, 90% is the best they can, if in a perfect world, is the best they can guarantee you. You always going to have 10% damage. So 1 in every 10 apple potentially have a strong.
Can I ask a question of Aaron? Your control method is very effective. Do you have any further comments on?
Oh, look,
Scott?
I don't normally speak too much, a bit shy, but like I've been processing all this and like I have got a bit to say. And for someone like Scott, who's got different sites, like what I've just seen and the data you show is like I can see a future to be using this product. And even if we could just freight it in at a cheaper cost, it'd be more cost beneficial than setting up somewhere to grow it ourselves, just so we can bring them off in. But my point is, it's like no one has trialled this moth without a site that's got pheromone trapping or port wine trapping or using an insecticide program. So you put that out there in a controlled environment, none of them, I reckon this sits, would probably take over the population of the apple gnawing codling moth, I suppose. So my point is if you can put it in an environment on a block or a site and use it only as that, I think you'd get a very good control. But like what's happening with what you have done is there's pheromone trapping, so we're catching them off. I know Mark Duggan's port wine trapping, we're still catching them off. It's not getting its potential outcome. And like, even if we're releasing it at the 60-day or the 60-hour period, it's weak. If we're going out and putting a sulphur or something on us, it's knocking this moth for 6. So it's actually not doing its full potential. So it needs to be on a site. Got nothing.
Those were 2 wet years where fungicide would have been key as well. So we're potentially knocking them a bit with the fungicide program.
It's like, how can you make this moth stronger when you put it on site? Like when you first go out and catch, like the biofix state codling moth, you don't ever take your first catch. You wait until you've got a good catch. It's like you can do your granulosis and product up until then, but it's like once you get your peak flight, like perhaps then's the time to release your SIDS and then back off your program and let it work.
Yeah, yes.
But like I've been a good believer of, and like even this year, not that I've got low labour on my orchard and that, but like even just letting the flowers and stuff like that happen in your orchard and you got to leave food there for the moth. You need beneficial like plants and stuff to help the moth. Yeah, stay strong.
Yeah, there's certainly some food for thought there, and perhaps it could be incorporated into the next iteration.
So the wet years, you say, are an issue for release. Is that exacerbated by ground release? Would aerial release be more effective on those wetting years? Actually get the moth up into the top of the trees. I think maybe it's too wet for him to climb up and keep slipping down. I don't know.
Just going back to 2014 or whatever when we went to New Zealand, but that was one of the reasons why they liked the hopper from the drone or the hexacopter is they reckon that dropping the moths in from that height woke them up and got them flying. Well, you know, they sort of released more passively in part of this trial, but yeah, who knows?
You know what time they done the releases?
Time of day.
Yeah, like late afternoon.
It would have been late after, it'd be just after the cappuccino.
Okay, I think the guy's got something to say. Sorry for not seeing your hand there, Guy.
Yeah, so I've been waiting a while, but that's okay. In the time I've been waiting, I've racked up a few different comments. I'll look, just start with that last one from Ian about the aerial releases. That's a really a good point and an interesting one, because what we observed is that the ground releases, you know, the moths appeared almost dead in the sense that they just didn't do anything, and it took them a long time to wake up. Well, once they woke up, of course, you know, they were active and flying, but they certainly weren't released, and they instantly started flying off. So, you know, aerial releases may provide, I guess, a more effective method of release if it does sort of wake them up and get them flying more actively. I know that in certainly in those early weeks, there was a lot of boggy ground. One of the sites was even flooded. I can't remember what it's called, had a particular name in reference to the flooding, but so certainly a lot of moths would have perished in those particular releases. So aerial releases might improve things. Just look, I say this not knowing anything about the economics of the apple industry, so forgive me if I've got it wrong, but even just looking at the sites we looked at, you know, the background level of damage is, at least some of the sites, is well over that 3.4% or whatever it was used in the economic analysis. So certainly, you know, if there are growers that are experiencing higher losses than that 3%, then that would certainly, I think, change the economic analysis and make it more viable. Another thing that might be considered, again, you know, I think growers are timing various control activities, you know, particularly sprays with regards to monitoring and trap detections. And so if that was potentially applied in the same way to sterile releases, and that rather than releasing every week for what is it, you know, five months plus, perhaps if you released more moths in those peak periods and fewer moths, or potentially even no moths in those periods where you aren't detecting moths. So obviously with this sort of very sort of short trial that we conducted, we had to release moths every week from very early until late. But you know, if you had a situation where you understood the moth activity and you were trapping to determine that each year, you could potentially tailor the releases through the season and reduce the cost that way. And the last comment I had, someone, I didn't hear it fully, but someone asked about I think the sterile releases, the effect on other insects, and I think I might have misinterpreted, because I think everyone else in the room might have thought that was about the effect on pests, but my reading of that question was that it was actually asking about the effect on other beneficial insects that might be predating on codling moth eggs or potentially larvae. And so I think in that sense, the sterile insect technique can certainly have a positive impact if it is resulting in a reduction of pesticide application through the season. So as well as your specialist biological controls like Mastris and trichogramma and others, you would have generalist predators, probably not doing a major level of control, but certainly they will be cleaning up some of the codling moth eggs. And so if you augment and build up those generalist predators and beneficials through a reduction in pesticide application, then sterile insect technology could have that flow-on effect of increasing the level of biological control in the orchard from other sources. Thanks.
How many days does sterile moth stay alive?
You go, Guy.
Oh, well, we didn't really measure that in the field. That would have been a fairly major undertaking that we didn't have the resources for. But in the control trials we did under laboratory conditions, basically most of them were dead after 2 weeks.
Being sterile, they're not going to lay eggs and reproduce. So yeah, so someone could bring them in on top of their spray program after and release them conveniently to suit the spray program, perhaps to attack the larvae left in their orchard.
Well, yeah, look, that's a really good question. I guess the key point there is that it's something that would be possible if we were producing our own moths here in Australia. When we're importing them from Canada, we have this very, very strict, very restricted, unlimited supply chain, so that basically we get them on the same day every week and they have to be released when we get them. Otherwise, their fitness just drops off and they become useless. You can't just sort of hold them over based on a grower's spray program. But if they were being produced locally or more locally, and you could perhaps tailor make, I guess, have almost like tailor ordering. You could maybe choose the day that you imported them for release. Then you could certainly incorporate them much more effectively with other controls that you're applying, because you certainly don't want to be releasing your moths and then spraying the next day. That's not going to work. But you could do it the other way around if you could control when you got the moths. But we obviously can't do that with importation from Canada.
There was a suggestion about Steritech. I did ask Ben Riley. He suggested that they didn't have the technology. It was inconsistent. Their technology was too specific. Now, whether or not there's about to be new investment in Steritech, but it was to do that at this level, he was feeling it was a far more specific treatment arrangement than they could handle. So there's not, unfortunately, it's not that just direct, easy, but it's worth a question given the he's easy to contact, worth a question to do. I guess
Yeah, but it wasn't my suggestion, Peter. It was what I said was that it's something that I've heard people mention and suggest. I have no idea whether Stereotech would be capable or not, but I have heard other people talk about it.
Yeah, sure, absolutely. And then I guess the other part was, so there's, I think then we talk about cherries, which have got very low pressure, but we do have this issue, a very costly issue in terms of codling moth trapping for cherries. And then we still got, you know, I know we didn't touch on the cost of codling moth trapping as a benefit, but also this issue we're shut out of, and CSIRO doing that work, we're shut out of Japan effectively because of our codling moth pest pressure, and we're required to fumigate. But if for some reason we can address that and then that comes into the whole systems approach, there is some real potentials. I mean, I can understand, especially when it's almost close to the doomsday clock, Sally, in terms of, so you can keep extending and expanding this, you must stick to within the lines. I guess it's just how do we finish off, and whether it's that desktop of where New Zealand's at. Guy's going to do the bit, which actually was going to go with Guy back in 2020, to go to Canada just to update about those sort of things, so we can just work out where to, so we can work out where next.
Yes, I think we can certainly expand our list of questions on our where to from here.
We could probably ask Jenny then to Lincoln.
Yeah, then she could add into that, Scott. You can do that.
Michael, we have another question in the chat. This one from Kevin Dodds. Is there a recommended time of day for release of moths?
There probably is a perfect time. I don't know what it is off the top of my head, but what I can tell you from this project is that because we are dealing, and potentially even with, maybe if they were, maybe less so if they were produced in Australia, but certainly imported from Canada, it's just not, it just wasn't relevant in the sense that, you know, we were racing against the clock to get them out while they were still viable. And as soon as we got them, we had to drive them down to the orchards and release them. And, you know, you can't sort of say, oh, well, we'll wait 6 hours for a better time of day. It's you just have to release them as soon as you get them, so that they are at their fittest and most competitive. There may well be an optimum time, but it's not something I know.
I mean, the reality is, Sally, I mean, this is not going to happen overnight.
No,
It's more a 5-year project.
That's right.
At the best.
It is a very
if
long
the industry
term
took it on as a whole properly, maybe the market would demand that we need to look at these things even harder in the future. But we're still going to be stuck with what we're using at the moment.
That's right, and it might get easier as more pesticides become unavailable as well.
And perhaps releasing them, you might need like a nice blower or a hair dryer or something.
I think shot of oxygen to come up a bit before they. And we've got a few abandoned orchards that maybe that's the place where we might release on cocaine. So, what was the overall cost, Phil?
The overall cost of
The work.
the whole project?
Yeah.
700,000.
Which is about what it costs to do your orchard to release once on today's costings for 120 hectare orchard.
But that wasn't, it was 90,000, about $90,000 a year.
Yeah.
To bring the moths in. The 700,000 was for everything that we did in the project.
Any more questions coming online?
No questions, just a couple of comments from Martin.
Do you want to pass those on to the group?
Sure. Martin noted that the rearing of codling moth is very different from fruit flies or blowflies as they do in South Australia. He does think it would require a dedicated facility and staff for that. He also notes that aerial releases allow usually to get a better distribution than ground releases. And for fruit flies, aerial releases are essential.
Well, I think we've covered a lot of ground today, a lot of sterile codling moth ground. And I'd particularly like to thank Guy and Sally for their presentations, and for everybody here and online for the excellent discussion, and to watch this space keep it in the front of your mind, keep asking questions. And thanks very much, everybody.